Welcome back. From the last class, we have started discussing about pollen-pistil interaction. Pollen-pistil interaction means here the pistil it accept only the compatible. Compatible means the same type of pollen grain. It allows only the same type of pollen grain. Here it is the structure of pistil. This is the stigma. A tube-like structure is called style, and the swollen part of the pistil is called ovary. This is the ovary. Here the pollen grain, they are come and fall on the stigma. That process is called pollination. That process is called pollination. The pollination, it can be occurs by the help of different agents. The pollination, it can be takes place by the help of different agents. It takes place by the help of wind, or it can be takes place by the help of water, or it can be takes place by the help of some animals like birds, insects and other reptiles, some primates. These are the agents of pollination. After the pollination, here the stigma, it accepts only the right type of pollen grain and it rejects the different species, means the wrong type of pollen grain. In this pollen pistil interaction, the pollen grain, it produces the tube. The pollen grain, it produce or tube and it germinate on the stigma. The pollen tube, it germinate on the stigma and it produce the pollen tube. That is the pollen pistil interaction. In today's class, I am going to discuss about double fertilization. What is double fertilization and where the double fertilization will be takes place. Here, after the pollen pistil interaction, the same type pollen grains will germinate and they are produce the pollen tubes. Here the most common type of oval, that is the anatropous oval. Here I will draw only one oval in a ovary, and these ovals are bitegmic. Bitegmic means they are having the two integuments, and anatropous oval means the microphylla region is present at the base of the oval and the chalazal region is present at the apex of the oval. Here it is the anatropous oval. Almost all the angiosperms, they are having anatropous oval. After pollen pistil interaction, the only compatible pollen grain, it produce the pollen tube. And here the pollen tube, it form. The pollen tube is formed and that pollen tube is enters into the oval. Here the pollen tube is enters into the oval. The pollen tube is enters into the oval. Here there are three possibilities of that pollen grain is enters into the oval. The first possibility, the entry of pollen tube into the oval by chalazal region. And the second possibility is the entry of pollen tube through integuments, and the third possibility is the entry of pollen tube through the microphylla region. Microphyll. There are three possibilities. Here the first possibility is chalaza, and the second possibility is integuments, and the third possibility is microphylla region. If this pollen tube enters through this chalazal region, the entry of pollen tube through the chalazal region is called chalazagamy. Is called chalazagamy. And if the pollen tube enters through integuments, these are two integuments, it means both the chalaza and microphylla. In between the microphylla region and the chalazal region, there are integuments are present. It means that is called mesogamy. Mesogamy. And the third possibility is the entry of pollen tube through the microphylla region. It means here that is called porogamy. These are the three possibilities of the pollen tube enters into the ovule. Here almost all the angiosperms, they are followed by porogamy. It means the pollen tube enters into the ovule through the microphylla region. This is the more possibilities. Here the pollen grain has two cells. The one is vegetative cell and another one is generative cell. And here they are having the two layers. The outer layer is called exine and it is made up of sporopollenin chemical. The inner layer is called intine and it is made up of cellulose and pectin. Here the sporopollenin chemical is absent in some regions, that is called germ pore. That region is called germ pore. Here the pollen grain it produce the tube. It means the intine layer, it produce the tube like structure through the germ pore. On that time the pollen grain has here the one generative cell. They are having only two cells. The one is vegetative cell and the second one is generative cell. When the generative cell when the pollen grain it produces the tube, means on that time here the generative cell it divides mitotically. Means one mitotic division takes place here and that generative cell produce the two male gametes. These are the two male gametes. The one generative cell it divides mitotically and produce the two male gametes. When the pollen grain it produce the tube, it means here this pollen tube has the one tube nucleus. Here the vegetative cell, it is also called tube cell. Here the tube cell is present at the tip of that tube and the two generative cell here it produce the two male gametes and these two male gametes are present here. These are the two male gametes. They are formed by generative cell and the one is vegetative or a tube nucleus and that is present at the tip of that pollen tube. This is the pollen tube structure. For our convenience, I will draw only the ovule diagram here. It means I will draw only the embryo sac diagram. Here it is the chalazal region. At the chalazal region the three cells are present and those three cells are called antipodal cells. The three cells they are present at the chalazal region, those are called antipodal cells. and the two nucleus they are present at the central cell, those are called polar nuclei. Those are called polar nuclei. And the one is large cell that is present at the microphylla region. This is the microphylla region, and that large cell is called egg cell. That large cell is called egg cell, and the two cells they are present at the adjacent of this egg cell. Those two cells are called synergid cells. Those two cells are called synergid cells. And this synergid cell has the finger-like projection. That finger-like projection is called filiform apparatus. Filiform tissue or filiform apparatus. Here, these all cells, antipodal cells, polar nuclei and egg cell, synergid cells, those all cells are haploid in nature. Those all cells are haploid in nature. Then, here the pollen tube enters through the microphylla region. Here only the pollen tube, I will only draw a pollen tube diagram here. The one is tube nucleus and that is present at the tip of the pollen tube, and here the two male gametes. These are the two male gametes. Two male gametes. They are present in the pollen tube. Gametes. This is the pollen tube. This is the pollen tube. This is the embryo sac structure. After that, the pollen tube enters into the microphylla region. Here, what will happen after? I will draw here. Here, the large cell is present. That is called egg cell. That is called egg cell. The two cells they are present at the egg cell. That is called, those cells are called synergid cells. Here the wall of the ovary. It is the wall of the ovary. And the synergid cells has the finger-like projection that is called filiform apparatus. That is called filiform apparatus. Then here the pollen tube enters into the one of the synergid cell. The pollen tube enters into one of the synergid or synergid cell. Why this pollen tube? Why this pollen tube enters into one of the synergid cell? Because of the synergid cell it has the filiform. The synergid cell has the filiform apparatus, and that filiform apparatus it helps to guide the pollen tube. That's why this pollen tube enters into one of the synergid cell. On that time, the pollen tube enters into one of the synergid cell, and this synergid cell disappear. This synergid cell disappear, and this pollen tube rupture, and these two male gametes are enter into the synergid cell. These two male gametes are enter into the synergid cell. Then the one of the male gamete it fuses with the egg. The one of the male gamete it fuses with egg. Here this egg cell is haploid. The male gamete is also haploid. Here this one of the male gamete, it fuses with egg cell. The first fusion here, the one male gamete, it fuses with the egg. Here the male gamete is haploid and the female gamete is also haploid. That male and female gamete get fused, they are formed zygote. The male gamete and female gamete, they are fused and they are formed zygote, and zygote is diploid. This process is called syngamy. That process is called syngamy. Here the first male gamete, it fuses with egg cell and the both haploid gametes are get fused. It means they are produced the diploid cell, that is called zygote. Here the egg cell, this egg cell become zygote. Then the second step or the the one male gamete it already fuses with egg nucleus, and here the one male gamete is present in this synergid cell. Here the one of the male gamete, it move towards to the central cell. Here it is the central cell. And the central cell has two cells. Means before the fertilization, these two polar nuclei get fuses, and that polar nuclei get fused and they are formed secondary nucleus. The secondary nucleus has 2 N because the one polar end and another polar nuclei, both two nuclei get fuses and they are formed the secondary nucleus. It means it has the 2 nucleus. And here the one remaining nucleus that is present in this synergid cell that move towards to the secondary nucleus. Here the another male gamete that is haploid in nature and it fuses with secondary nucleus. The secondary nucleus has 2 nuclei and they are formed the primary endosperm cell. It is called PEN. PEN means primary endosperm cell. The secondary nucleus, the one of the male gamete, it fuses with the secondary nucleus, already 2N is present. The one of the male gamete fuses with secondary nucleus. It means the 3 nuclei. The one is male gamete and the secondary nucleus has 2 nucleus. It means the primary endosperm nucleus has 3 nuclei. This process, the fusion of 3 nucleus, that process is called triple fusion. That process is called triple fusion. Then here the one male gamete, it fuses with egg cell and it forms zygote. And that process is called syngamy, and the one male gamete, it fuses with the secondary nucleus. It means the 3 nuclei get fuses. Here the secondary nucleus become primary endosperm nucleus. That fusion is called, or that process is called triple fusion. Here the double fertilization process. Double fertilization means the both processes, here the syngamy and the triple fusion, these two processes takes place in a single embryo sac, that is called double fertilization. Double fertilization means the syngamy process and the triple fusion process takes place in a single embryo sac, that is called double fertilization. Here After the double fertilization, the zygote, the first is zygote is formed. First zygote is formed, and that zygote it becomes embryo. Zygote it becomes embryo, and here in secondary nucleus the one male gamete fuses and that secondary nucleus become primary. Secondary nucleus become primary endosperm nucleus. Later this central cell, this is the central cell, and that central cell become endosperm. The central cell become endosperm. Sorry, the central cell become primary endosperm cell. The central cell become primary endosperm cell, that is called PEC. PEN means primary endosperm cell. PEC means primary endosperm cell. Here the zygote, this zygote become embryo, and the central cell, here it is the central cell, and that central cell become primary endosperm cell. In double fertilization, in embryo sac, the one male gamete is haploid, egg cell is also haploid. The one of the male gamete is fuses with the egg nucleus and it forms the zygote. That process is called syngamy. And the next second male gamete, it fuses with the secondary nucleus. The male gamete has haploid nucleus and the secondary nucleus has two nuclei, and these two get fused and they are formed primary endosperm nucleus. That fusion or that process is called triple fusion. Why it is called triple fusion? Because of the three haploid nucleus, they are get fused, and that fusion is called triple fusion, and they are formed primary endosperm nucleus, that is also called PEN. After that, the central cell of the embryo sac, it become primary endosperm cell. Primary PEC. PEC means primary endosperm cell. After double fertilization, the zygote it become embryo, and the primary endosperm cell it become endosperm. The primary endosperm cell become endosperm, and this endosperm is the nutritive tissue. It is the nutritive tissue. And this nutritive tissue, it helps to or the developing embryo. This developing embryo, it nourishes by the endosperm, or the endosperm it nourishes the developing embryo. Here the double fertilization means the syngamy process and the triple fusion process. Both two processes takes place in a single embryo sac is called double fertilization. This is about the today's class. And what are the significance of double fertilization, or what are the advantages of this double fertilization? Here the significance of double fertilization. The first significance is double fertilization, it results in the formation of zygote and primary endosperm nucleus. After double fertilization, the zygote it become embryo, and the primary endosperm nucleus it become endosperm. Here the second significance is the endosperm. It is the nutritive tissue and which helps to nourishes the developing embryo. The nutritive tissue, it helps to nourishes the developing embryo. And the third one is the fertilization. It is necessary for growth of embryo and it leading to the fruit formation. After fertilization, the ovary it become fruit. And next one, the zygote is formed. The zygote is formed by the fusion of male and female gametes, and it helps to maintain the constant number of chromosomes. These are the significance of double fertilization, and this is the today's class, double fertilization and the significance of double fertilization. Thank you.